Hello, everyone, and welcome to Agulhas Current Affairs, a marine science podcast set in Southern Africa, with me as one of your hosts, Dr. Janus Kirsten of Geekoscopy Media, alongside
Dr. Nelson Miranda of Argonaut Science. And today we're going to get bold. We're going to go for it. Yeah.
Oh, jeez.
Yeah, there's going to be a lot of acronyms. A lot of acronyms today.
A lot of acronyms. Oh, I thought you were to get confident about something.
Well, yes, that too, of course.
That too. Okay, let's fight the introversion today on this episode with DNA somehow.
Yeah. So, have you ever heard of the Barcode of Life?
I have heard of the Barcode of Life. It does come up in undergrad when you learn to become a marine scientist. But I didn't myself go too deeply into genetics. I've worked with DNA more in a physiological sense. But Barcode of Life, I understand, it's got to do with DNA and it's got to do with biodiversity and kind of tracking those things. Am I in the right ballpark here?
Yeah, you're totally in the right ballpark. And I mean, it's a barcode because, you think about a barcode that you find on a product that you might buy at the supermarket. And it's got those lines, dark and light, and it makes a barcode. And instead of lines, what we're talking about today is the code that is encoded in every organism in the animal kingdom. And really all things that live, they have DNA, which is like kind of the building blocks of the information from which life is built, right?
Yeah.
And very cleverly, there's a very specific gene, so a very specific bit of DNA that is very common it's common to all respirating life, life that breathes, things that are generally considered to be alive. And it's a gene that all genes have these funny names, right? So it's the cytochrome C oxidase subunit 1 gene.
Yes.
And that sounds very
sounds like you just turned into like a robot,
Cytochrome C oxidase subunit 1.
Yeah. Executing order 66. Yeah.
Oh my gosh! Don't do that.
Don't do that.
It's too soon. No. It's normally we just call it CO. We abbreviate it CO1 or COI because we use the Roman numeral 1. That's why it looks like an I.
Oh, yeah.
So, and that that sequence, okay, it's a really suitable one for a barcode of sorts because it's common to all forms of life. But it does mutate, right? So DNA mutates over time, and if it mutates too much, that's not so good. If it mutates too little, then it's like preserved, and that's also not great because then, how could you tell things that mutate in sort of species as they mutate, they can actually become separate species the more they mutate, So that gene, though, is like perfect in terms of the mutation rates over time, and also it is passed on, throughout, from the generation to generation because it's a very important gene, because it's the gene that is literally it's needed to code for the protein, right? That then does the job of completing that that very important process in respiration where the electrons move, electrons in energy, and they get packaged into something called ATP. Anyway, let's not get too... ATP is like the power fuel of cells, and this is the power of life, right? Yeah, but we don't have to get
think of it
too
like,
technical.
when you're...
DNA.
Think of ATP as money, right? And it's your like energy source, and you're at the grocery store and you're trying to figure out like how much money you need to pay for a thing. Like all of the products have that barcode, and you take it to the front, and they scan it, and they tell you like how much it costs and how much you have to pay for. So that's kind of like what's going on here, I suspect.
Well, there's actually a link. I like the mind map linkage, and I like to think, yeah, ATP could be like the moolah for life. But the most important thing to focus on are those barcodes themselves, which are made up of different codes of DNA sequences. So DNA base pairs they called, and they can either be A's or C's or G's or T's, right? So those are the codes. So to go T T A C G T T T A, so it's like almost like boop, 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 little code of sorts. Almost
Sure.
like Morse code. But in this case, there's actually different sort of elements, and that's DNA. That's how DNA can be used. And that barcode lives, it lives in organisms. So it's, no one has to put it on. It exists in every organism, right? So now the idea, right, is to, extract that DNA from organisms in a process that, I think we'll cover this in a future episode about DNA, where we can go into it a little bit. And DNA extraction, isolation, and then sequencing, right? Isolation and we were talking about this earlier. The CPR and CPR.
PCR, yes.
PCR.
Yeah,
PCR
chain reaction.
Amplification. Polymerase chain reaction. There you go. Now who's a robot? So we can go into that another time. But basically, you do need an organism. You need to isolate the DNA. You do need to extract it. You do need to purify it. You do need to sequence it. So you do need specialized tools, which you can find in universities, and there are companies that have these labs where you can send samples. And this can be done for you, and then they will give you a sequence, one of these sequences with lots of A's and T's and C's and G's, right? And that sequence is based on like what you've set it up to be. So we use something called a primer, which is like it's going to be the key that isolates out of all the DNA exactly only the CO1 gene. That is what we're interested in, that little sequence that's going to be our barcode, and then it's going to give you like a whole bunch of codes, right? And then what do you do with that? You need to put it in somewhere, into a database of sorts, isn't it? And you have basically two big options, right? There's an open source option called GenBank, right? So you can bank it in the GenBank, right? And then there is what we're going to be talking about today, the Barcode of Life systems, Barcode of Life Database system, right? Or BALD. You can get BALD. And you can do both, actually. And the difference really between the GenBank and this Barcode of Life or BALD system is that the GenBank is open source, and anyone can input like all of these sequences, and there's not as many checks, just to make sure that everything is correct. There are some, for sure. It is still a process to submit. It is published. It is acceptable in publications as well to deposit these sequences into that bank and then reference them. So you get the unique reference. And you can say, there's my sequence that I collected. And then scientists can actually mine that and extract it and compare notes and make sure that, if they return to where you found that organism and they sample again, they're going to find the same thing, right? That's quite important. But then the BALD database, right, system is a step further. It's curated, you know. You need very specific photographs. You need definitely the GPS location, you know, where you found this organism. You need taxonomic identification already, right? Or the closest that you can get. You need the actual trace files, right? Which are these technical files that you'll get from the company that sequences your data. They'll give you these files, and you need to actually input those, and they need to read forward and backwards on your code, right? So all of that needs to be there as proof that everything is kosher, and you didn't just like type in some random things together. So yeah, there's a lot of more checks. So it is a bit more complicated to get it right. But once it's right, if there's something in there, in that database, then you can be pretty confident that it's a solid database. And at this stage, you might be wondering, why are we going through all of this trouble? You know, all of this.
I was about to ask, how does this relate to marine science? Because this
right?
is marine science podcast.
Wow, because there's so much. It's so technical, and you can't just like grab a scanner like you do in the supermarket and just go beep. You know, it's not so easy, right? Why bother? Well, because we simply can't keep up with you know. There's so much life out there, and especially in the ocean, there's so much to discover, right? That if we were to use conventional methods like we've been doing for centuries, right? We would take even more centuries. We'd probably never arrive at the destination of trying to identify, you know, all life. You know, trying to know what life exists on Earth. And well, this is one of the best tools. It's even though there's a lot of work, it is quicker and it is more reliable, right? And this can really help solve a lot of problems, you know, problems of identification. You know, taxonomy is something that's always changing. And there might be misidentifications. Things might look the same even in terms of shape and size and color. But when you look at the DNA, then you start to find, okay, it's actually the one code is mutated and is kind of moved away from the other code, and it's actually almost like a subspecies or maybe two different species that look very similar. So we can use these approaches to really simplify and help us to even take care of some of the messes. You know, there's a big mess in always with naming things, and so it helps.
That gives me an idea. We should probably have an episode about what is a species. Because I think it's quite a complex thing that we might need a more modern way of trying to explain. But it's not so straightforward these days what a species is.
No, exactly, and it's still debatable. And now even with these tools, sometimes how divergent the sequences need to be for something to then say now it's a different
where do you draw the line?
There's always going to be an element of where someone has to come in and decide what thresholds are. And there's also, a history. So someone has to come in and look at the past and things that have been put into museums and need to be revisited, readdressed. And then how do we then, be fair to the original people that found these things, so that, because authorship and, ownership and discovery is also important. So there's a lot of things to consider here.
How does this fit in for South Africa? What is South Africa doing in the context of barcoding?
Okay, so there's something very exciting going on right now. There's a project called CMAP, right? And this is a big countrywide project, right? It is based at SAONS, that's the South African Environmental Observation Node, the Egagasini node, I believe, and this is a big project through the Foundational Biodiversity Information Program, or FBIP, right? The FBIP program funds huge projects in the country. There's another one, for example, that I'm also part of, is the Refresh project, where we're looking at biodiversity in the freshwater aquatic realm. But there's a little bit of salinity that goes in there as well. So don't discount freshwater people from the marine people, by the way. But this FBIP program, these programs are funded by the Department of Science and Innovation, so DSI, with the South African National Biodiversity Institute, that's SANBI, and the National Research Foundation, NRF. Those SANBI and NRF, they manage these projects, right? So the CMAP aims to deliver South Africa's first data-driven marine ecosystem map, right? So in the Barcode of Life is very important. This is how we actually measure the progress of this project. The Barcode of Life is part of the outputs of this project. So basically, this project is using the Barcode of Life to map and contribute a huge amount of knowledge about marine life in South Africa.
Oh, that's pretty cool.
Yeah.
Ecosystem map derived from associated species. Yeah,
absolutely
amazing. Yeah.
So then, if you want to find out more, also the principal investigator is Dr. Lara Atkinson, and you can find out more if you Google on the internet, and there's a lot more information available. This is like hot news, eh? So we were in a meeting, a few weeks ago, where most of the scientists involved in this project came together, and you must realize that these are scientists from different fields or different, specialists of different organisms, different, that's fish specialists and invertebrate specialists, and and algae and microorganisms as well, bacteria, all in one room, trying to figure out how can we work together. So this is very exciting stuff.
Yeah, for sure. It's sometimes very difficult to have like multidisciplinary, even big teams getting together to do a single project. So
absolutely
expect good things.
Absolutely, the community is great, and scientists are actually working together to trying to figure out exactly those issues of how, because the method of extracting DNA is going to differ, wildly, between taxa, but then lessons learned from one side of the field may be applicable to the other side, and we may come up with a standardized way of collecting this information that serves everyone the best that we can. But yeah, it's a very exciting thing that's going on. But you know what? There's also, if you go and you look at the Bold Systems portal online, right? So Barcode of Life Systems online. There's even an opportunity for you as an educator, as a teacher, to get students involved in this project, because this is an international project, right? The BOLD project, right?
Yeah.
And if you do it in South Africa, of course, I definitely would recommend that you liaise with other scientists that are working, on this front. So the CMAPS project would be, people to approach there. But for sure, you could definitely, benefit from some of these tools, which will guide you in, learning how to barcode, even as a school teacher. And definitely, you would need to collaborate with a scientist, because you definitely need guidance and you need access to facilities, which a scientist, for example, is working on the CMAPS project may be able to help with.
For sure, 100%. I think, definitely go check that out. I think we threw a lot of information at the viewers today, so maybe we cut it off.
I mean, it's exciting, but we have to be careful not to overload the senses.
It's an overload.
So
I am a scientist,
we'll probably cover DNA and something called.
So yeah, I think definitely go check out what's going on with CMAPS. Check out the educational portal. It's something that's interesting to you guys. I'll post a couple of interesting, entertaining things about DNA in the description. I do know a couple of songs that exist about DNA and PCR, and I'll find some other entertaining related things, and we'll probably discuss more about that in a separate DNA episode. And we'll have an episode about species, because that's its own topic that needs talking about. But otherwise, I think it was a very interesting topic that we'll probably check up on later on.
Yeah, again, we're probably going to cover DNA and something called next gen sequencing, like eDNA and metabarcoding in another episode. eDNA is really exciting because instead of finding organisms, you can maybe sample water, and because there's fragments of organisms in the water, you can actually extract DNA and then do this kind of barcoding as well. So really cool stuff for the future.
Excellent. So yeah, if you want to find out more, I think you should go head over to Nelson's social medias at Argonaut Science. Where can people find you, Nelson?
And you can find Argonaut Science on the web. Just look for the octopus, Argonaut Science.
Cool. You can find Agulhas Current Affairs at Agulhas Current on Twitter, or Agulhas Current Affairs on Facebook. You can find me at Geekoscopy Media, and I do post these at geekoscopy.com for you guys to listen to that. So otherwise, stay tuned and cheers.